Here with us today, Harvard is a postdoc in between. That's true. I can tell you that he does beautiful work. I know his older work on the conduction system that was really beautiful. And more recently, it's really more about the genetic basis of congenital heart disease. And just this morning, I read a paper in Nature that came out of your lab, and you probably hear about some of it today. And it's really striking, in particular, the effect of voluntary, and I'm quoting, voluntary exercise, which means involuntary exercise doesn't work. Voluntary exercise by the mother that can mitigate predisposition for congenital heart defects. I know that the work was done on Ph.D, but maybe others too. Okay, so without further ado, it's a pleasure to have you.
Thanks. So the picture that was shown to announce the seminar is from my last paper on the conduction system. We had this observation. So as a postdoc, I studied the role of this transcription factor, NKX25, in the development of the conduction system. And at the time, the field thought that any defect related to conduction in the postnatal heart must be related to a defect in an ion channel gap junction, basically proteins that propagate the action potential. And so I showed that how the wiring is laid down, a developmental problem, could affect conduction. And after that paper was published, there were a whole series of examples published to show that development itself could affect electrophysiologic function. The cells themselves are intrinsically normal. And so when I first came here, we wrapped up the experiment. The idea, and I tell you this story in the hopes that maybe someone will understand what the result means and can carry it on, because it's probably important. I'm not smart enough to know how or why. We paced we crossed mice that had an NKX25 mutation. Those mice and humans have fewer cells in the central and peripheral conduction system, half as many if it's a heterozygous mutant. We crossed those mice to mice that expressed GFP in the central conduction system. It was driven by a GFP reporter. And that's the picture that you see in the seminar announcement. So you see the atria lit up, the AV node, and the His bundle. And what Avi Hugazit, who's now a cardiac intensivist, and Alex Lee, who is now a general surgery resident, discovered, is that if they paced, so we flayed the heart open so we could expose the AV node and see it with GFP. If they paced the superior aspect to the AV node, they could get conduction. As they increased the pacing output, the stem to H interval, stem to His bundle interval, would shorten, I think, in the wild types. On the other hand, we never saw that property in the mutants if we paced it from the inferior approach. So the conclusion of this little paper was that there's something intrinsically abnormal about the cells in the inferior approach to the AV node. In the mutants, we have no idea what it means. So we just published the observation. After I started the work and moved to St. Louis, I realized that the field of the cardiac conduction system, at least as related to development, was getting crowded really rapidly. So, and I didn't want to deal with the competition. We had to pick a new problem. I decided as a cardiology fellow, so this was really a long time ago, that basically the field of cardiac development was going to have no impact on the management of congenital heart disease. So what is congenital heart disease? There are 4 chambers in the heart. The veins send blood to the atria, either from the lungs or the body. The arteries, the atria convey the blood to the ventricles pump blood to the lungs or the body, right, through the arteries. Any rearrangement of the plumbing that you can imagine has been described. So the veins don't connect to the right place, the arteries don't connect to the right place, valves don't form, there are holes between chambers don't form. And the problem with that, and so the cardiac developmental biologists have been really very good over the past 20 years at figuring out which genes in the embryo are important for regulating cardiac development. So I'll give you a few examples. A couple of years ago, with the advent of whole exome sequencing, this consortia of giant pediatric cardiology centers around the world have collected now almost 10,000 patients. With their first 1,000 or so, they collected trios, so the baby and the two parents. And their simple question was, are there mutations, de novo mutations in the baby that are associated with congenital heart disease? And as their starting point, they had a list. They surveyed the big shots of cardiac developmental biology, and they came up with a list of 300 genes that were involved in cardiac development. And I show them here. This is supplemental table S12. And actually, that guess is actually pretty good, because now that there have been many sequencing projects in both humans and mice, the estimate really is that there are probably about 300 genes that can be mutated to cause heart defects. All these genes are in the embryo. It is not obvious to me how any of them can be manipulated to prevent a heart defect from forming in the first place. So the way congenital heart disease is managed is we react to the fact. The heart completes development by the 10th week of gestation. We cannot diagnose a heart defect, so we can only diagnose serious heart defects by the 18th week of gestation. So for serious heart defects, the game is already over, right? We're into plumbing mode. So what you want to do then is, if you want to prevent disease, you want to intervene long before you even know that the heart has finished forming, or the defect has formed. You can look at it from a mouse angle. So a few weeks ago, there was a paper published in Nature, a giant project where this lab did ENU mutagenesis. What ENU mutagenesis is a chemical is given to the males that causes mutations in the sperm. The mutations are crossed to recessiveness. The details are not important. And what they discovered from scanning, I think, almost 150,000 mouse embryos, so they just put an ultrasound probe and scan to look for really bizarre defects shown here. So the top two panels show a normal 4-chambered heart, and the other 6 panels show examples of double outlet right ventricle, where both arteries come out of the right ventricle, or there's a big AV canal defect here, where the atria and the ventricles are connected by a common valve. So this group found 5 genes involved in cilia. So cilia are little like hairs on cells that push signals left, right, that detect signals, molecules floating around, right? And so these have also been implicated genes. So there are a bunch of genes, all of them in the embryo, none of which I think I can fix. And I think the investigators would be hard-pressed to explain how they're going to fix hearts based on this. So through this graph continues. So what I did, and to make this case clear, is look at the number of papers published related to 3 prototypical cardiac transcription factors. So it was discovered in the 90s that mutations of cardiac transcription factors cause congenital heart disease. And these are the first 3 discovered, TBX5, GATA4, and MTX25. So molecular biology starts about here. And you can see that there are a huge number of papers that contain these terms and/or cardiac development. And the number continues to grow with next generation sequencing strategies. This list is really huge, right? And the papers are just exploding. On the other hand, this explosion of so we're talking about inside the box. This is where the field is right now. This explosion of basic scientific knowledge, I would argue, does not translate to improved clinical outcomes. So here are the number of deaths per 100,000 children in the infants by birth in the United States as a function of time, right? And so before 1970, basically if a baby was born with a severe congenital heart defect, that baby was going to die. With the advent of advances in surgical techniques, you see this curve start falling. And then around here, I think the curve is beginning to level off. The reason why I think there might be a little bit of a drop is not because, I would argue, because of tremendous advances in technology. Those occurred about 20 years ago. It's because of the diffusion of knowledge and best practices from the big centers to everyone else. So this is the history of science and technology, right? Everyone catches up. And so outcomes are slowly leveling off. Science is picking up. these two are clearly not going together. And it's not clear how these 300 genes are going to inform management. The improvements in surgical care of children with congenital heart disease is really just temporary. So what this graph shows is even though we can keep the babies alive, right, often now until adulthood, we only delay the onset of problems. So there are now more Americans with severe congenital heart disease, adults, than there are children. So that's true. I think that surveys have been done in the US and in Canada. So in the developed world, that's striking. And this paper from the Brigham and Women's Hospital a few years ago basically looked at rates of hospitalization for congenital heart disease. So in the small black bars, you see there are going to be more and more adults. In about 2020, and the number of children being born with congenital heart disease is constant, roughly 40,000 a year. So in about 20 years, there are going to be more children, in about 10 years, there are going to be more adults in hospital with congenital heart disease than there are children. So it's not going to be my problem anymore, it's going to be Dr. Reginald's problem. Right? And so what we need to do, I would argue, is think outside the box to figure out how to bend the curves. Right? So I need to bend this curve. I need to make the science matter for congenital heart disease in terms of affecting outcomes. And the best way, I think, to do this is to think about prevention methods. And that's what my lab has worked on for the past 10 years. To begin, we need a mouse model. And I picked the one that I got from my postdoc from Seigo Uzuma's lab. We have a mouse that has a knockout of a gene, NKX25. NKX25 was the first gene discovered to be associated with non-syndromic congenital heart disease. It's a gene, a transcription factor expressed in the fly hearts and Drosophila. When you knock out that gene in the fly, no heart forms, and that's why it's called tinman. The mouse was made in the hopes that it would replicate the fly phenotype. But I think to the chagrin of the two labs that made it, the mouse did develop a heart, even though it was not normal. I think work would have stopped there had it not been for the discovery that humans that have heterozygous mutations of this gene also have congenital heart disease and AV block, which was the subject of my postdoc. And so that caused us to re-examine the heterozygous mutant mice. I'll show you examples where basically they have the exact same heart defects that humans have. And this presented now an opportunity when I started my lab in St. Louis to take congenital heart disease outside the box. The basic idea was that we have mice within that carry a heterozygous mutation of NKX25 in an inbred strain, C57 black 6. What an inbred strain is, if you don't know about them, is that every gene, every base, every copy within the mouse is identical. And what that means is within an inbred strain, all the mice are genetically identical. But between strains, they are different. Right? And so if you want to study the effect of a genetic mutation within the strain, you know that the difference between your mutation and, say, wild type is related to the gene itself, not to some unknown genetic background effect. I discovered as a fellow that mice in this genetic background had a high incidence of heart defects. When we crossed it to one of two inbred mouse strains, FEB or AJ, almost none of them in the F1, so it's half black, half white, has a defect. So they're all basically normal. This suggested that there must be other things in the genetic background that influence the development of a heart defect. So the NKX25 mutation is necessary. The wild type animals are all normal, but there must be something else, right? So there could be additional genes in this mouse that lead to the manifestation of the defect. There could be protective genes here that suppress the effect of this mutation. The other possibility is that there are, protective and susceptibility alleles in both backgrounds, and they just happen to complement in the F1, with the protective ones being dominant to the susceptibility alleles, right? To figure this out, we cross the F1 mouse to either the parental strains or to itself, and just looked at these three crosses. So you have to remember your high school biology and think about recessive and dominant. But basically, we found that in all three crosses, we recovered mice, mutant mice that had heart defects. So that means that there are protective and susceptibility alleles in both strains that come out, that become recessive again here to cause heart defects. And so that suggested to us that we would be able to find these genes. And the hope was, if we found those genes, I may not be able to fix the NKX2-5 mutation or one of the other 300 genes that have been worked on, that are being worked on in the embryo, but maybe these other genes could suggest a pathway that we could target to prevent congenital heart disease. To do that, we needed to set up a really very efficient mouse cardiology clinic, because basically what we're talking about now is epidemiology in a numbers game. It's incredibly hard to find genes by conventional genetic linkage analysis. It's a statistical game. And to get good statistics, you need to increase a sample size. So just for reference, we probably collected now, I'm sure we've collected now well over 30,000 mice in my lab. And if you come by, we just have shelves of boxes of mouse hearts. And we have a giant database where every single mouse is tracked. So to set up a cardiology clinic, what we do is we collect newborn mice daily. We have all their DNA. We section them, like here. We phenotype them. So the hearts are sectioned like a loaf of bread or a CT or MRI scan. The sections, every section is arrayed on a slide. And then we have a team of students. I can train a high school student, someone who's never been to medical school or even taken anatomy, to be as good as the first-year pediatric cardiology fellow at diagnosing mouse heart defects. We need this. We need this type of system because attending cardiologists are really expensive, even more expensive than postdocs. And then once we phenotype them, we can associate them with SNP genotypes for linkage analysis. This is the team in session now. So Yor mentioned a Nature paper that just came out. Half the authors were undergraduates who helped phenotype the mouse hearts. And some of them helped with the statistical analyses too. For the cognoscenti in the group, this is a mouse with tricuspid intrusion. We see simple, so the top two panels show a wild-type mouse heart with normal 4 chambers. We see all the types of defects seen in humans. So this is an example of a membranous VSD. You see the septum coming up like a finger, and then it doesn't close at the base of the heart. There's a gap here. This is a mouse with a muscular VSD. So that's a hole between the left and right ventricle and the muscular septum. This is a mouse with an atrial septal defect. Blood can flow between the left and the right atrium. We also see complex defects. This is a common atrioventricular canal. This is an example of a mouse with a double outlet right ventricle where both great arteries, the aorta and pulmonary artery, come off the right ventricle. Complex defects are most practically important because that's where the money is, right, in pediatric cardiology, and that's where all the excess mortality is. We're actually very good at fixing simple septal defects, and the outcomes are great. So a few years ago, as we started doing this experiment, we developed data to basically show the world that we were on the right track, that we would find genes that would affect the risk. So this is a very simple diagram that shows in various genetic backgrounds. I think the backgrounds are not important. You can see that the incidence of various defects varies, as if genes contributed by a particular mouse strain steered the heart towards a particular type of defect. So, for example, atrial septal defects are more common in this cross than in this cross, and common AV canal defects are more common here than here. I told you that we have DNA for every mouse, so it's possible to statistically associate genotypes or loci with specific heart defects. There's a black mouse and there's a white mouse. We have markers throughout the genome that basically can distinguish the black and which parent gave which allele of a gene, right? And then from there you could do statistics, linkage analysis, to figure out where the important genes may lie for steering the heart towards a particular defect. And that's really what happens here. It's as if there's a cookbook, a combinatorial cookbook, where in the presence of an NKX2-5 mutation, genotypes at various loci push the heart towards a particular defect. And it's a very quantitative approach to congenital heart disease, really very different from the way developmental biologists or cardiologists think about it. So when a baby presents to me in clinic, either the baby has disease or not, right? But here, we look at a particular mouse, yes, it's a binary outcome. Either the mouse has a defect or not. But when you look at a population, it becomes a statistical problem, and we're looking at it in terms of the probability that a mouse with a particular genotype will develop a defect. It's not a binary problem anymore. So for membranous VSD, the defect where there's a hole at the base of the heart, in the ventricular septum, we see in this particular cross loci of chromosomes 6, 8, and 10. And the probability is quite high that there is a gene in these intervals, on these 3 chromosomes, that affects the risk of developing a membranous VSD in the presence of an NKX2-5 mutation. The genes that steer the heart towards one particular defect are not the same for each defect. So here is an example of a muscular VSD, and this is really very early data. We just see one peak on chromosome 6 that might be shared, but when you look at chromosomes 8 and 10, there's really nothing. The map looks completely different. And since then, we've increased our sample sizes, and this has been a consistent theme. Every single defect has a different genetic map. Whenever you talk about genetic linkage analysis, it's basically a pit that, a huge genetic mess. The reason why it's a mess is typically when you do mapping, you can never map to the causative gene, right? And so even though these peaks look really nice, right, there are still 100 or more genes in the interval. And for a molecular or developmental biologist, it's incredibly hard to figure out what those genes are. And what I realized very early on in this, in looking at the history of labs that do quantitative or do genetic linkage analysis, is if they cannot jump from a big locus, so a wide interval, to a gene, they do one of three things. So they must find a new disease to map, they must find the gene, or they must shut down the lab. I have 4 kids, and they're still really little, so I can't shut down my lab. And so along the way, we've been trying to figure out ways to do what I call jump to a gene. Ahiakarome, an MSTP in the lab, is just a font of ideas. And this is an idea that's really not quite ready for prime time, but I throw out there for you guys to think about, and maybe you will have some input. He had this idea, and he's starting to do experiments now, where maybe all these loci are somehow functionally related to each other, right? And even though there are hundreds of genes in total between, say, in this map, 3 loci, maybe the gene that underlies each of them is somehow functionally related. And then he had the simple idea that maybe, because these are mice that have just one normal copy of NKX2-5, maybe there are genes that increase the activity of NKX2-5. And he started looking at all our data with this hypothesis in mind. And he's now found, I think, at least 3 loci where that's probably true. So in this locus, there's a gene called FOXP1, where expression in the endocardium inhibits the expression of NKX2-5. So you can imagine that there are strong or weak alleles that have this effect. And let's say if the repressive effect is more, the mutant that carries the NKX2-5 mutation plus a strong FOXP1 allele would be more likely to have a defect. A weaker FOXP1 allele, or maybe a mutation, might cause paradoxical increase in NKX2-5 activity. FOXP1 is regulated by a gene, the FGF family of genes. FGF20 sits on chromosome 8 and induces or regulates the expression of FOXP1. This gene is in the endocardium, and there's a growing literature now about how the endocardium talks to the myocytes to regulate cardiac development. And what you can imagine then is that failure of growth of that muscular ventricular septum, or poor growth, prevents it from reaching all the way to the base of the heart. And he has found another example for muscular VSD, and a SNP on a microRNA binding site that regulates another key transcription factor, islet 1, that's involved in the switch from cardiac progenitor cells in the second heart field as they go from outside the heart into the heart. There's a switch regulated by islet 1 and NKX2-5. And I think that's probably not a coincidence that that SNP is in the conserved microRNA binding site, also known to be important in cardiac development. We haven't discovered the genes yet, but from these type of data, you can infer the biochemical activity of whatever the gene is from looking at the patterns of inheritance. So for example, on chromosome 6, if a mouse has 2 copies of the C57 allele, the mice are more likely to get a heart defect, a VSD in this particular case. So it's that whatever that gene is, and perhaps it's an enzyme, has dominant, it has recessive activity, whereas having 1 copy of FF is dominant of FF. And so here, the heterozygous looks equivalent to this. Make sense? And then you can make the same argument for this locus on chromosome 8. And this one has a pattern of codominance, where the heterozygous is intermediate. So you can imagine an enzyme that has a combination of activities. And somehow, one is not better, C or F is not better than the other one. They have equal strengths. Excuse me. So I told you that a genetic mutation in combination with polymorphisms at genetic loci steer the heart towards specific defects. We have a pretty good handle now on simple septal defects. I think we're making progress in understanding what those genes might be by taking non-genetic approaches. So either just really educated guesses, bioinformatic approaches, fancy genetic strategies. The other thing that we're very interested in is just thinking about severe heart defects in general. And this might be important in thinking about how we might approach management, the management of disease. So if you buy my argument that genetic modifiers steer the heart towards a particular defect, you might wonder, can we utilize that to understand and maybe fix rare defects? And so I have a very simple question. Why is it that severe defects are rare, right? So the defects that are most likely to kill you are the least common. So in children's hospital across the park, they're incredibly common, but in the real world, they're not. For the purposes of writing grants, they're a public health menace. In reality, really not that big a deal, right? It's a big deal for families. There's still a fair amount of money spent on taking care of those children. And so, can the genetics inform how we think about disease? So there are a couple possibilities for thinking about this. NKX2-5 mutation, genetic modifiers, AD canal, or some other horrible defect. Are the defects rare because there are many genetic modifiers such that it's highly improbable that an individual gets the right combination of modifiers to get that heart defect? So it's kind of like a combo shot in pool, right? If you have to hit 5 balls just right to sink the last ball, you're never going to make that shot. The other possibility is that there are just a few genes involved, and there's something about them. It's a low, even though you have the right combination of genotypes, it's low penetrance, or there might be alleles that are strongly protective. So we can answer this question by just mapping this defect. So in the plots I showed you for simple septal defects, we had to map hundreds of mice. But in this particular case, it only took a few dozen to show that just 2 loci, probably just 2 loci, explain the development of common atrioventricular canal in NKX2-5 mutation. So that suggests, because we needed very few mice, that it had strong effect. And if you look at the, this is another way of looking at the effect plot, it's quite clear that if a mouse has a protective genotype at either locus, the mouse will either not get a defect or will be highly protected from getting a defect. So Blue represents common atrioventricular canal, red represents normal. And what you notice here is that in red, it follows the typical expected Mendelian distribution, so 25%, 50%, 25%. Do you remember that from high school in Punnett squares? If you cross two heterozygous parents, right, you will get 50% of them will be heterozygotes, and then 25% of them will be homozygous for either allele. Right? On the other hand, if a mouse here has a CC genotype on chromosome 3 or an AA genotype on chromosome 5, that mouse is not going to get an AV canal defect. So what that suggests then is if I knew what that gene was, and if I could mimic the activity of that gene, I could prevent a severe defect in that mouse, even if it had an NKX25 mutation. I might not be able to present an ASD or a VSD, but I've already told you that that's easy to fix. So Ahi, in the lab, he has a background in evolutionary biology. He's begun to wonder whether there's a general pattern related to the evolutionary origins of congenital heart disease. An atrial septal and the effect on the strength of selection on genetic modifiers. So you would expect then that if there are modifier genes with protective alleles, the strength of protection would be really strong for defects that kill you right off the bat, because evolution selects on survival of the species, right? And so the insurance to make sure you don't get a bad heart defect should be really big. And so the hypothesis is for ASD, the protective alleles that we find should be relatively weak. ASDs do not kill you until middle-aged, so 40s in general, or older. So anyone with an ASD should survive to reproduce. On the other hand, VSDs are intermediate, so if it's a big VSD, those patients will die as children generally. If it's a small VSD, they will not die. If it's an AV canal defect or other type of severe defect, those children will die as babies, so they will never live to reproduce. Ahi plotted the data for all the loci that we have mapped, and basically the pattern is that if a mouse, it's a weird way of thinking about the math, but basically what he does is he looks at mice that do not have a protective allele, and if they do not have a protective allele, their risk for severe defects, so AVSD at top, is significantly higher than the mice that have simple septal defects. And there's a pattern, a trend like this. We're expanding this, and I'm pretty sure that for other defects like double outlet right ventricle and AVSD and other genetic backgrounds, we're going to see the same basic pattern. So evolution selects on the strength of these polymorphisms in the modifier genes. The strength of protective alleles is related to the severity of defects. So it makes sense. We can explain congenital heart disease and the patterns of incidences that we see based purely on evolution. We don't have to invoke like the cause of congenital heart disease. So this has nothing to do with the cause of congenital heart disease. This is all about the genes in the background. So what these data show, and if you look at all the literature and interpret the incidence of congenital heart disease for various types of diseases, say like Down syndrome, where there are 3 copies of chromosome 21, it all begins to make sense. This is a trick question that I throw out to doctors and students. So in Down syndrome, when you have 3 copies of chromosome 21, I will ask them, what's the most common cardiac diagnosis? And the good students and docs will say, AV canal defect or PDA, something like that. Okay? And I say, wrong. The most common cardiac diagnosis is structurally normal heart. So that's really incredible. You can take a big hit to your genome. There are mechanisms to keep you on track. And I think this is what our data are telling us. So I think disease exists on a bell-shaped, or actually a multi-dimensional distribution. For simplicity, I draw it as a bell shape, where disease exists on the ends. Most of us live here. So if a cosmic ray, a tragedy, or mutation hits us, what happens is that in a population, most of us will have modifier genes or buffers that push us back to the middle. You need buffers that go in many different directions because you don't know a priori which way this cosmic ray is going to hit you. It could push you to the other way, right? For this mechanism to work, there are going to be some people who get dealt the equivalent of a genetic royal flush. So this is, I think, really very interesting and has, I think, ethical implications for why we should take care of people who have disease, not just congenital heart disease, but all disease in general. So I suspect that basically all of us here are normal because of this system, and evolution necessarily sacrifices some people, right, so that the rest of us can be normal. They just happen to get dealt a bad hand that once they got hit with the cosmic ray caused them to veer off course. It's not just congenital heart disease, so a textbook example of this is sickle cell disease. Right? So sickle cell disease relates to mutation of hemoglobin. If you have one bad copy, you're protected from malaria. If you have two bad copies and you live in the United States where there's no malaria, you get sickle cell disease. There was a great article in New England Journal of Medicine that argued that basically all diseases of the red cell relate to selection pressure by malaria on the human genome. Right? And the reason why we see disease in the Western world is because these patients basically offer their genome so that the rest of us would not get malaria. So I've told you about genetics. The other hat I wear is as a doctor seeing patients in clinic. And so cardiologists and surgeons are very practical people. We are all about intervention. We are not generally into philosophizing. We just want to get things done, get people out the door. So how do we translate this information? When I first started, I had a very strong genetic bent, but the first graduate student in the lab said, maybe it has nothing to do with genes. Maybe there's something else in our colony that affects the risk of heart disease in our mice. And I thought, that can't be true. Right? But since it cost us no money, I let her do the analysis. So I told you we have a database, so every day of the year, we go in the morning and we collect babies, collect their hearts and the DNA, and we have excellent we have complete demographic information for every mouse that's born. We know its mother, its father, the day it was born. We know the day that the mother and father were born. We know all its brothers and sisters. And so she just ran a simple logistic regression analysis on the factors that might vary from the perspective of the embryo. So as the embryo sits in the womb and develops, the embryo, we thought, can see its mother, the age of its mother, right? It knows how many brothers and sisters it has around it in the womb, and it knows the age of the father. And to our surprise, the age of the mother came out as a significant risk factor. So what we plot here is the incidence of one particular type of defect as a function of maternal age from age 48, that's the youngest mouse we have, we have in our colony, to over 500 days. So this is about 4 or 5 weeks of age. That's the equivalent of a mouse adolescent teenager. And when a mouse is about 1 year of age, it's very hard to say what it is in human years, but I estimate it's around age 40. We scratched our heads for a while trying to figure out what this means. Because we have DNA on every mouse, we quickly ruled out that it was chromosome aneuploidy, so Down syndrome. That would be the trivial explanation. Right? Down syndrome is associated with aging. The thought there is that the eggs go bad as the mother ages, and the chromosomes aren't able to separate normally, so the baby gets 2 copies of chromosome 21. Right? And with the father's contribution, there are 3. So I don't show that data, but basically, aneuploidy is not going on in our mouse population. We look through the human epidemiologic data and realize that this is actually a consistent observation. So we just, at the time, pulled all the papers that looked at the effect of maternal age, even after eliminating cases of chromosomal aneuploidy, and it's a consistent result. Maternal age is associated with the risk of congenital heart disease, but not always. And so this got us really excited because now I told you, if I can't find a gene, I'm going to have to shut down my lab. Right? This opens up the possibility that there's something else to study, something else to target. And we could run the math in a logistic regression analysis to look at the effect of genes or aging on the risk of congenital heart disease. So this is something that Claire Schulte did. So on the x-axis is plotted risk factors. So a mouse could have no genetic risk factors or a risk genotype on 1, 2 chromosomes or all 3 chromosomes, and as a function of maternal age. So genetic risk remains constant over time. That's a one-time hit. But as the mother ages, that risk accumulates, right? And so in very old mothers, so in 400-day-old mothers, the effect of aging can be as big as, or bigger than the effect of genes in the embryo. And so if you want to, place your bets, you should study aging to have an impact. And so the question is, what is the basis of the maternal age effect? Most people would say that it's bad eggs. That's received wisdom, right? The eggs are formed when the mother was an embryo, and they just go bad. I don't know how, right? But if you look through the literature, the evidence is really very circumstantial that it's bad eggs. The other possibility is that there's something in the mother that affects embryonic cardiac development. Actually, as mothers age in humans, the risk of many different types of birth defects goes up. And so we did an experiment that, as far as I know, no one has ever done before. You could do it in a mouse, you could never do it in humans. In humans, with epidemiology, you can only get associations, you can't get mechanisms. So what Claire did was transplant ovaries between young and old mothers. So here's an old mother and a young mother. We just put the ovary here and vice versa. And then we can look at the incidence of heart defects as a function of the age of the ovary or the age of the mother. So we've collected data now on thousands of mice, and so we can calculate what the expected incidence is. So for either a maternal or an oocyte basis. And basically, what this shows in the observed, so we collected the babies of these mothers, is that this matches almost exactly that expected for a maternal basis of a maternal age effect. It's significantly different from an oocyte basis. That's really exciting because it is much easier to develop therapies for an adult, the mother, than it is for an embryo. I already told you that the heart defect has already formed even before we can diagnose it, right? And even if an embryo or a one-celled fertilized egg has a genetic predisposition, you don't know if it's going to develop a heart defect. So it's really very hard to figure out who you should treat. So we started thinking, what is it about, there must be a factor in the mother. I define factor very broadly that affects the risk of a heart defect and that interacts with cardiac development in the mutant. There is no age-associated risk in the wild-type mice. So it's the same story. You must have an underlying genetic predisposition in the baby for other factors to influence outcome. So there are a couple things associated with aging and maternal aging and congenital heart disease. Obesity and insulin resistance or diabetes are among them. The data are quite clear for that. So what we did was we asked, can we accelerate the maternal age effect? Is the maternal age effect related to something simple and already known? We put the mice in a high-fat diet. At an early stage, they become glucose intolerant. At a late stage, they become very obese. This shows that we can make them very diabetic. So this is a glucose tolerance test where we give the mice a slug of glucose. Normal mice very quickly can go back to the normal baseline levels, whereas mice in a high-fat diet, they have this huge excursion in peak glucose. It takes them a long time to get back down to baseline. The mice become morbidly obese, so at age 40 weeks, they're more than twice as heavy as normal mice. On the other hand, the high-fat diet has no effect on the age-associated component of risk. So in this, we just divide it simply into young and old mothers in this particular graph, right? So in black is the baseline incidence with mice fed a normal diet, and white is mice on a high-fat diet. It doesn't make the offspring of young mothers worse. In the offspring of old mothers, there's a trend, but it's not significant. And it's certainly not as big as the effect on the mother in terms of diabetes and obesity. And then so if you look at the entire population, we can calculate the risk associated with it. So basically, if the risk is greater than 1, it's positive. If the bar does not overlap 1, that means it's statistically significant. And a high-fat diet does not significantly increase the odds associated with maternal aging. And you notice here we look at thousands of mice. If it's not diet, obesity, or glycemia, what can be? So we needed other clues. And I told you that we've looked at crosses in different genetic backgrounds. We've taken C57 mice, cross it to FVB or AJ, take the F1 and then cross the F1. So we quantified the maternal age effect in 3 different strain backgrounds. Either the 2 intercrosses or pure C57 black 6. And what we showed excuse me, is that the maternal age effect is a quantitative genetic trait. There is a pathway in the mother, genes in the mother determine the activity of some factor that affects the risk of heart disease. So it's very high in pure C57 black 6, it's intermediate in C57 FVB, and it's non-existent in the cross of C57 AJ, and the difference between the high and low is significant. This last cross is telling, because C57 and AJ have been studied for decades for their sensitivity in C57 and resistance in AJ to the effects of a high-fat, high-sugar diet. So even though I told you that diet did not matter in our experiment, metabolism is really very complex. And we surmise that there's probably something more than just sugar and fat that underlies the differences between these 2 strains. And so what we did instead to try to get at the problem was a rescue experiment. The idea is, maybe I don't know the genes or the molecules that are important, but if I can prove that I can prevent heart disease, that would put us on the right path and keep us going in terms of studying this maternal age effect. So the question became, might exercise help? If you look through the literature, for many diseases of aging in human and mouse models, making them exercise has clear effects, beneficial effects. The mechanisms are not necessarily known. So we had mice in 3 different groups. So sedentary mice, that's our control. We had mice start exercising early at age 4 weeks, as soon as they were ready to reproduce. And at a late stage, 8 months. But what we discovered was that exercise eliminates the effect of aging in our mouse model. So in black, our offspring of young mice, right? And here's sedentary, here's old. If you look at mice in the old group, if they've exercised, if the mothers have exercised basically all their lives, and what this exercise is called voluntary, because all we did was put wheels in the cages. They're the exact same wheels that you find in the pet store for your kids. Mice love to run. It costs like $4. Mice love to, it costs a little more because we had to buy stainless steel ones that can be autoclaved by the animal facility. The plastic ones melt. They love to run. If you go into our colony, you will always hear squeaking wheels. This has been studied. So if you put mice in a cage with a wheel, they will run, I think, up to 16 kilometers a night. So it's incredible. Mice have really short legs. All right. And so we basically, with early onset exercise, reduce the age-associated component of risk. And this is a huge reduction in risk from about 20% to 10%, right? If you could eliminate half the congenital heart disease in Children's Hospital, Dr. Agdasani and I would have to work a lot less hard than we do. It'd be pretty awesome, right? I knew, though, that it would be impossible in terms of intervention, to make women exercise beginning when they were the equivalent of teenagers. So the question is, can we start it later in life? And so that's why we did the 8-month onset experiment. And basically we get the same reduction in risk in the incidence of VSD. VSD are shown because that's the most common defect. It's very hard to get statistics on rare defects. That's a statistical power. But I think the trends are probably there. And then when you do the odds ratio calculation, basically eliminate exercise makes the risk associated with aging insignificant. A modest amount of exercise is sufficient, right? And so what this data shows is the late onset exercise experiment. We asked how many days does a mother have to exercise before you can see a detectable reduction in risk? And basically the answer is a mother has to exercise about 3 months prior to conception to eliminate the age-associated component. So it doesn't work if they start during pregnancy, right? It has to be about 3 months. So I don't know what it is, but if we want to do an experiment, we have to set to find the molecule. We have to let them go for a certain amount of time before we can purify that molecule from the blood. The mice, and I say it's modest because the mice don't have to exercise so much that they become like svelte supermodels, right? That's really very important. And I think that's consistent with much of the adult literature on the effect of exercise in cardiovascular disease. You don't have to ask an older adult to become really buff, right? To reduce their risk of coronary artery disease and a heart attack from that. Just some exercise, a few times a week has a significant effect. So we looked at glycemic indices, basically did the glucose tolerance test again. The mice don't appear less diabetic, right? The normal mice don't have disease, so it's not really less diabetic. They don't appear healthier in terms of their insulin sensitivity. And then we put the mice in an MRI machine to quantify their lean and fat body mass. And the mice are not more muscular. They're not. They don't have less fat. The exercise and the sedentary mice look basically the same. So exercise does something really pretty subtle in terms of affecting the molecular biology and molecular physiology of the animal. It's not something obvious. And when you think about it, it makes a lot of sense. I told you that diabetes and obesity are associated with the risk of congenital heart disease in humans, but not every mother who has diabetes has a baby with congenital heart disease. So I suspect that what we're looking at is a statistical association related to a subclass of mothers. So the association of, say, diabetes and congenital heart disease, that's true. But really, there's probably some small fraction of mothers that have diabetes and some molecule related, say, diabetes and inflammation, or diabetes and compound X, right? And it's the compound X that's associated with diabetes that affects the risk of congenital heart disease. And probably the same thing here, so we don't affect diabetes or body mass or obesity, but we're affecting compound X, and that's the key thing that's important. That's what we want to discover.
Does the effect relate to the age of the mother, so that the older the mother is, the more you exercise is required?
So we haven't done that experiment. So that's where all the money is in terms of like if you want to do human trials, like you're basically saying, is it too old and it's not worth it anymore to exercise, right? The other question, and this is what we're getting into, is my prediction is that exercise will not be beneficial for everyone. So if there's a mother who's like the AJ mouse, there's no amount you can, because she doesn't have an age-associated component of risk. There's no amount of exercise that's going to reduce the risk for her baby. On the other hand, if a mother is like C57 with a high thing, you really want to make those people exercise. And the key then is how do you identify C57-like humans and AJ-like humans, right? And this is where I think the mouse research can really inform future clinical trials. So what my lab is doing now is trying to basically narrow the search space to make clinical trials more efficient. And that's my goal. By the time I retire, I want there to be an intervention or a pill that we can give, right, to prevent congenital heart disease. So I probably if my R01 gets refunded, right, gets renewed, I expect to work for another 20 years, right? If not, well, this is the difference story. But you know, most drug companies now, they don't want to prescribe drugs for everyone in the world, right? They don't want to do a clinical trial because if they get a negative result, they've just spent $1 billion to prove nothing, even though they're pretty sure that there's a subset of people whom the drug would benefit. And so they want to find those people beforehand to show a positive result in their clinical trial and then get FDA approval, right? So what we can do here is, in this particular case, is an advanced intercross. I showed you the F2 intercross experiment where we can map big chunks of DNA that contain hundreds of genes. You can set up a cross such that the chromosome, instead of being, say, half black and half white, gets cut up like a zebra. And so there are very fine genetic intervals. And we can map, instead of, say, this mouse here, right, where the chunks of DNA are big, really these mice here. So we have various crosses from the, say, the 10th generation up to the 55th generation of intercrosses. At the 55th generation, we can map down to 4 genes. And if there are only 4 genes in an interval, I can begin to do biochemistry, developmental biology, whatever. I can open up a textbook because if there's some factor in the mother that's transmitted across the placenta, there's probably a lipid, some small molecule. Big proteins generally do not cross the placenta, right? Biochemistry is really very well understood, so I can just open up the biochemistry textbook and see what gene is important in that interval. What gene is listed? I know nothing about biochemistry, right? But if it's lipoxygenase in those 1 of 4 genes, that's a clue. And so that's what we're doing here, is to try to map to fine genetic intervals. There are actually, so I have this view that science is very Hegelian. There are ideas permeating the universe. Scientists do not come up with ideas. Ideas pop into people's heads simultaneously around the world at the same time. And I just came back, and so then it becomes a race into who can publish the idea first. So Hegel, I call it Hegelian, because Hegel is a philosopher who said, I think basically he said, men do not make history, history make men. Is that right, Elliot?
Sounds right.
Yeah. And so I just came back from a meeting where this human group tried to look for maternal genes that were associated with congenital heart disease. And their #1 gene was something called ABCG1. So something that transports lipids across the membrane. And I thought that was really very telling. The other thing that we're trying to do is, that we're gearing up to do, is metabolomics. So you can imagine, we've spent many years now looking at risk profiles. So I have high and low risk strains. I know exercise makes it better. You can imagine then, say, collecting the blood from mothers who are young and old, exercise or not, and then by comparing the metabolites in the serum of these mothers, you can define a small set that are associated with the risk of congenital heart disease. They might be protective molecules or susceptibility molecules, depending upon their hypothetical mechanism. From here, we can cross-reference this list of molecules to our genetic data, right? So if it's a lipid, there should be a lipid enzyme. We can also do a conventional experiment where you have a pilot study and then a validation cohort. So this experiment is ongoing now, where we're basically quantifying the maternal-age effect in 18 different lines of mice. And so if there's a molecule here, you might expect that the concentration of this molecule should be related to the size of the effect in these strains where there is an effect, and the molecule should not be present in strains that do not have the effect. So in conclusion, I've told you briefly about inside the box thinking, so that's where most of the field is, looking at direct causes of congenital heart disease. My lab for the past 10 years, and a growing number of labs are really buying into this model of modifiers, whether it's genetics in the embryo, in the mother, or environmental factors that, in addition to the direct causes, steer the heart towards normality or to disease. These modifiers, I think I've shown you, can suggest a way to prevent congenital heart disease. So I may not be able to make every mother in the world exercise, right? But if I could put exercise in a bottle and make everyone buy it, pretty awesome. They can be genetic or environmental, but it depends upon your perspective, right? From the perspective of the embryo, maternal age is an environmental factor. Defining the properties of the modifiable pathway in the mouse can inform human studies. So there are roughly 20,000 genes in our genome. If you do a human genome-wide association study, if you follow this literature, say you want to discover genes for myocardial infarction or QRS interval, you need to collect 100,000 humans before you can find a little signal, right? Congenital heart disease is relatively rare. There are only 40,000 children born a year in the U.S. with it. We are never going to collect all 40,000 in one fell swoop. It would take forever to collect enough patients to find genes. But if you could narrow the search base in a mouse experiment, then we can start identifying, are there mothers who have molecules associated with aging and the risk of congenital heart disease? And then if you identify those high-risk mothers, we could then target interventions at them to detect a benefit. And then that's the point of the last conclusion. We've shown in the mouse model that we can target a pathway. It's not just wishful thinking. There's a practical example now where we can actually prevent congenital heart disease, at least in the mouse, and proven principle that you could translate those results to human. So to do this work, required a large number of people in the lab. These are the people currently in the lab. For the undergrads are Megan, Herman, Rachel, Ida, and Mahi. These are all students who help diagnose or do statistics in the lab. I showed you the work of Claire Schulke and Julia Winston, and Suk Regmi. He's a technician in the lab. He sections all the mouse hearts. So without him, the lab would not exist. And these are people I collaborate with in various projects that I've told you about. These are people who pay for the work. Thanks.